Mercer will speak about category theory for genetics.
Okay, so I'm going to talk about a work that is due to Rémy Tuyéras. I don't know if I'm pronouncing that correctly. He's at MIT with David Spivak. Okay, so this talk's based on a series of papers written by Rémy. So the first one, category theory for genetics, this is the one that I'm going to talk about today. You can find this one on the archive. And then there's another one with a very similar title. This one is called Part 1. This one was recently published in TAC. Its journal neighbor is a pretty cool paper, open Markov processes. And then there's another one, Part 2, that talks about some different stuff. So I'm mainly going to talk about this first one today, the first half of that. Okay, so these papers are meant to set up a categorical language that's suitable for talking about things like DNA sequencing, alignment methods, Crispr, homologous recombination, genetic linkage, and lots of other things. So I'm definitely not going to talk about all of these today. I'm mostly going to talk about the first one and a little bit about the third one. And then all these other ones are covered in the remainder of the first paper as well as the other two. Okay, so first we need to do a little bit of biology background. So our bodies are made up of cells, and these cells duplicate when they undergo a process called mitosis. So here's a very vague picture of a cell splitting apart into two daughter cells. Okay, but before a cell can undergo mitosis, it needs to create a copy of the DNA which resides in the nucleus. So the DNA is basically like the cell's instructions on how it's supposed to function and do stuff. Okay, so the DNA replication happens before the cell undergoes mitosis. So roughly 90% of the cell's life is spent in this phase, not necessarily replicating DNA, but in this pre-mitosis phase. I think this is called interphase. And then roughly the last 10% of the cell's life, it undergoes mitosis and duplicates. Okay, so our DNA is made up of a sequence of nucleobases. So these might sound familiar. So we got cytosine, guanine, adenine, and thymine. And so the only thing that's really important for us is going to be the letters C, G, A, and T. And so these nucleobases are interwoven into this double helix shape. So I don't know if the picture is too fuzzy, but these nucleobases are in complementary pairs. So guanine goes with cytosine. So in other words, if there's a G over here, there needs to be a C on the other side, and conversely. And adenine and thymine are complementary pairs as well. So if there's an A over here, there needs to be a T over here. Are you guys able to see these letters? Okay, so that's roughly what DNA looks like. Okay, so before the DNA is replicated, it needs to be cut open or unzipped by an enzyme called helicase. So here's a picture of the DNA being cut open. I think I have a better picture of this on the next slide. So after the helicase comes in and cuts open the DNA, then these primer enzymes called primases, they bind to the unzipped strands, and then these other guys called polymerases come in and they start copying. So here's a picture of all of that. So one thing you'll notice, or at least I noticed, is that basically anything that ends in ASE is an enzyme of some sort. So we've got primases, we've got polymerases, we've got helicases, and yeah, so if it ends in ASE, it's some sort of enzyme. Okay, so here's the helicase going along, unzipping the DNA, and then these polymerases, they come in and they start copying. So this guy's job is pretty easy because he can just follow the helicase because they're both going in the same direction. This other guy, he has to work a little bit harder because he's copying stuff in the direction opposite that this thing's being cut open. So he needs to copy this little piece, and then he needs to come around, copy the next piece, come around, copy the next piece, so on and so forth. So this is called the lagging strand of the DNA.
Why does it have to go that direction? Why couldn't it be smarter and go the other way?
So I believe it needs to be copied in the 3' to 5' direction. So this guy goes 3' to 5', and then the other strand goes 5' to 3'. So that's why it needs to go backwards.
So there's a directionality built into these single strands.
Yeah.
And I guess you'd need to have two different kinds of polymerases if you wanted to invent one that went along backwards, so to speak.
Yeah,
nature didn't want to do it that way, so they made one that goes sort of in the same direction, but then it has to like jump around back.
Yeah, it's actually not the same. It's not a single polymerase that does all this copying. It's a bunch of these guys on this lagging strand, and it seems like there would be a lot of room for error copying this guy because you got a bunch of these guys coming in. But then there's some other stuff that happens to make sure things are as it should be. So. Okay, so then after the DNA is replicated, then the cell undergoes mitosis. Each one of these new cells grabs one of the original copies of DNA, and then the cell splits apart, and you get two cells like that. Okay, so another thing that our DNA is used in is RNA transcription and translation. So in this process, we have another enzyme called Rna Polymerase that comes in, it binds to the segment of DNA, and it opens up the DNA, but only internally inside this enzyme. So here's a picture. So it's not cutting the whole thing open like the helicase was. It's just cutting it. It's just opening it up on the inside, and it's transcribing one of these strands, and it's creating this mRNA transcript. So I think I have a better picture on this slide. So one thing to notice is that this RNA strand, so this guy's got U's for uracil instead of T's for thymine, but its nucleobases are still complementary to the ones on the DNA strand. It's just using U's instead of T's. So okay, so we get this strand of mRNA. So before it exits the nucleus and goes outside into the other parts of the cell, it has certain parts called introns that need to be cut out of it. So that's these blue guys. So these get cut out, and then this is what exits the nucleus. So that guy leaves the nucleus, and that's the end of transcription, and now we're ready to begin translation. So outside of the nucleus, this strand of mRNA. So first of all, this thing's made up of little 3-letter sequences like this called codons. These ones in particular, so AUG, this is a start codon, which signals for the strand to begin translating once it's outside the nucleus, and then these other 3, these are stop codons. So outside the nucleus, the strand gets this ribosomal unit attached to it. Okay, and as this guy goes along the strand of mRNA, so if you look at these little guys right here, so this says U, A, and C. Okay, so it wants to find the letters that go complementary to these guys. So I don't know if you can see the shape, but so A goes with U, U goes with A, and G goes with C. So this guy in here wants an AUG, and attached to an AUG is going to be some sort of amino acid, like these 2 guys right here. So then this guy moves along, goes to the next 3, and then the little amino acid in here gets attached to this growing chain. And so this thing is building this chain of amino acids. So here's another picture of that. So this guy just keeps trucking along until he gets to a stop codon, and then this guy disassociates from the strand of mRNA, and then this blue necklace looking thing, this is the end product of transcription and translation. This thing goes off and does good stuff in the cell and in the body. Okay, so that's the biology review. So now we're gonna do some actual math. Okay, so for every positive integer, let n in brackets denote set 1 to n together with the implicit ordering on the positive integers. All right, so let omega be a preordered set. So a segment over omega is going to consist of a pair of positive integers, n1 and n0. It's going to have an order-preserving surjection from n1 to n0. Okay, so in particular, this guy needs to be at least as big as that guy. And it's also going to have a map going from n0 into the preorder omega.
Is that last one order preserving?
No, that's just a function. That's not order preserving. Okay, so what all this means, so n1 is the number of nodes. That's what I'm going to refer to n1 as. n0 is going to be the number of patches. This is all very visual, and I'll have an example in a second. n0 is the number of patches, and this order-preserving surjection is going to group the nodes into patches. So T basically is going to give a segment its topology. And the function C is going to say how each patch is to be interpreted or read. So here's an example. So if T is going from 7 to 3, then one example, what T could be something like this. Okay, so T is just going to take these 7 integers and it's going to group them into three patches. Okay, so this is a patch, this is a patch, and this is a patch. So I'm not going to denote them with numbers. We're going to use little black dots like this. Okay, so here are the nodes, and they're being grouped into patches according to this order preserving surjection. So that's what T does, and then we also have this map C, which is going to tell us what we're supposed to do with each of these patches. Okay, so in about 98% of this talk, I'm going to take omega to just be the Boolean preorder. 0, 1, false, true, white, black. So for example, if I say, okay, I want C of 1 to be false, so I'm basically going to be using the colors. So white's going to be false or 0, black's going to be true or 1. And if I define C according to this, just for this particular example, then what this is doing is it's coloring the first and the third patch is white and the second patch black. So if I combine this with T, then really what this guy is going to look like is this, okay? So I got 7 nodes, I got 3 patches, and the function C specifies what each patch looks like. Okay, so I think I already said this. So T gives the segment its topology, and C gives meaning to each patch as specified by the preorder of omega. So the notation for a segment is just going to be a paired TC. I'm going to draw it like that.
So far you haven't used the preorder in omega at all.
Right. So most of the time I'm just going to be using the Boolean preorder, but omega can be something more complicated. So I think that's what this example is going to be about. So remember in RNA translation, before the strand of mRNA exits the nucleus, it needs to have those exons cut out of it. So we could have a preorder that looks something like this, where the green and the red, those are our start and stop codons, and we ignore the blue, we ignore the black and we read the blue. So we could have segments that look something like this, where these are the guys that we don't care about. Start here and there, and we care about the blue ones.
What idea is that preordering?
It's just an example. I'm not going to be using this one for the rest of the talk. It's going to be the Boolean preorder.
Are the blue and the black like the intron and extron?
Exactly, yeah.
Like, does that preordering have a physical interpretation?
Say that one more time?
Does the preorder up there have a physical interpretation?
Does it have a physical interpretation? I mean, the physical interpretation. I mean, it's basically as Christian was just saying. So before this guy, if this represents a strand of mRNA, before this thing leaves the nucleus, we need to get rid of these introns. So, I mean, that's as much as I could give you for a physical interpretation.
But that doesn't say like why read is above ignore. The post-set structure is what's bugging all of us. On the preorder structure on omega is what's bugging us because you don't seem to be using it yet. And in this example, it's not apparent why read should be above ignore or above terminate.
Seems like the colors just come from C. I don't get what the diagrams do.
Well, that's showing the pre-order structure. But anyway, so far, we would all be just as happy if omega was just a set. Because nothing you've said uses the less than or equal to structure on omega.
Maybe as I go further, that will clear itself up. So later on, I'm going to be taking omega to be the Boolean preorder, and it's only the black patches that I'm going to care about, and the white ones I'm going to disregard. So that's really all I can say about how the preorder is going to come into play. I don't know if that answers your guys' question.
What is a preorder group?
What is what?
A preorder?
So it's a set equipped with a binary relation that is reflexive and transitive. Right. So we have two segments over omega given by these two guys, then we can talk about maps between these two segments. So a morphism from this first segment to the second segment is going to be a pair of maps, F1 and F0, where F1 is going to be an order-preserving injection from N1 to N0. F0 is going to be just an order-preserving function from N0 to N0, such that we get a commutative square like this. And we're going to have another condition that C prime on a guy in here is less than or equal to C of a guy in
Oh, now we use the pre-order.
there. What?
Now we finally use the pre-order.
Yeah. Okay, so now I want to talk about what morphisms look like in some special cases of F1 and F0. Okay, yeah, so we get a category. That's nice. So the objects are given as segments over omega, which are pairs, and then the morphisms are, as I just said, they're these commutative squares where F1 and F0 are order preserving or whatever you want to call it. Okay, so what can we do with this category, and what do its morphisms really look like? So the rest of this talk, omega is going to be the Boolean preorder, even though I'm going to mention it about 10 or 15 more times. So one thing we can look at is what Rémy calls locality. So if we have a patch and we take F0 and F1 to be identities, then the only condition, so let me draw, I want to put this little square patch over here so that I can refer to it as we go along. So N1, N0, this is TC. So this guy's an injection, this is F1, this is going to be N1 prime, N0 prime, this is F0, so this guy commutes. And we also have that C prime of F0 of I is less than or equal to C of I for all I in here.
Just for fun, write C prime down on the bottom arrow.
Happens over here.
No, on the bottom arrow there.
I see what you're saying.
And we really, those horizontal arrows were both sort of
Yes.
rejections. Yep.
Okay, so some special cases of this are when F1 and F0 are identities. So when F1 and F0 are identities, really the only condition we have is that guy right there, aside from the square commuting, but now, whoa. Yeah, so if F1 and F0 are identities, that's the main condition that we have. So what that condition is saying is that the colors in the segment are going to need to decrease, or more specifically, they can't increase. So what I mean by decrease, this is where the preorder comes into play. So 0 stands for white and 1 stands for black. So we can only go down. So we can only go from black to white, or we can stay black. We can't go in the opposite direction. So here's an example of a morphism of segments where the F1 and F0 are identities, and I'm making this patch, which is initially black, I'm making that white. This guy is staying the same. These two are being made white, and all the white ones stay the same. So in other words, I can't, if this guy's white up here, I can't make him black down here. Okay, by that condition.
So is changing colors saying this has just been read or just been copied and we're done with it, and now we're changing priorities to...
Yeah, it can be. Yeah, depending on how you're really interpreting false and true. If you're taking that to be like ignore and read or something like that, then you could give it a more precise meaning than what I just said. So that's one thing. Another thing that we can consider is what Rémy calls relativity. So if we relax the condition that F0 be an identity and we let that be whatever we want, but we keep F1 to be an identity, then we can merge patches together like this. Okay, so in this particular example, I'm taking these three patches and I'm merging them to this patch. I'm merging these two patches to this patch, and I'm merging these two patches to this patch. Okay, so again, the color decreasing condition comes into play. I can't merge these three patches into a black patch here unless these two guys were also black, okay? Because there's whites here, when I merge these three patches, I have to go white, okay? Because I can't increase their color, okay? This patch, they're both black, so it's fine to merge them into a black patch. Okay, so we can merge patches together.
So F0 has to be surjective in this situation. At least in your example, it's surjective, right? So
Yeah, in general, it doesn't have to be. And later on, there's going to be an example where F0.
It might have to be surjective in this example, this type of example, right? Because F1 composed with that other thing is, oh no, never mind, because F1 doesn't need to be surjective, never mind. Yeah. So you drew one where F1 was surjective.
So F1 here should be a bijection because I'm not changing the number of nodes.
Right. So we didn't. Yeah. So the diagram didn't force you to not change the number of nodes.
No, yeah,
But
just in this
one
particular
case
example.
decided.
Yeah.
Okay. Yeah.
Well, there's a lot of things that you can.
Yeah, I was just trying to sort of see like how much of this example is like forced to be sort of the way it is by the conditions and how much it's just the victim example.
Okay, I think I got one more little buzzword, flexibility. So what this means is that we can increase the range on patches, okay? So here, I'm not changing the number of patches for either segment, but here, the F1 is an injection, but it's not a surjection, okay? So this patch is going to this patch, not everything in here is getting hit. This patch is going to that patch, so on and so forth. So I'm just increasing the size of each of these patches. So that's what some of the morphisms look like. They don't all look like this, and later on we're going to see some different kinds. So this is just to give you an idea of what the morphisms look like. Yeah. So in this particular example. Yeah, because the number of patches is staying the same, I'm taking F0 to be an identity. It doesn't have to be an identity. And I'm also supposing that none of the colors actually decrease, which is just saying that the colors of each patch remain the same. It doesn't have to happen. Okay. So if we have two segments and their topologies are the same, then we say that those are homologous. So we've already seen an example of this earlier. So that was this example. So
T is just what the topology is just your name for T.
Yeah. I'm calling T and T prime the topologies of these two respective segments. Okay, so in other words, it's saying that the number of nodes doesn't change and the number of patches doesn't change. The only thing that changes is and if we fix a T, so in other words, if we specify a topology, then we can look at segments that have that particular topology. And if we take the F1 and the F0 to be identities, well, that's going to preserve the topology as we go from one segment to the next. So then we get this subcategory, segment of omega T, I don't know whatever you want to call that. And this guy is actually a preorder category, which means that there's at most one morphism between any two segments. So that's kind of nice. We can weaken our conditions a little bit and say, okay, I don't want T and T prime to be the same, but I want them to both have the same domain, which means N1 and N1 prime have to be the same. So we say those are quasi-homologous. And we've already seen examples of that. So this is the one where I'm merging the patches, but so in this particular example, the number of nodes stays the same. So these guys are quasi-homologous, and if we fix a positive integer, then we get another preorder category where the objects are segments with N1 nodes in them, like these two guys right here. And the morphisms, we're going to take F1 to be an identity so that we preserve the number of nodes as we go from one segment to the next. Okay, so that's a couple preorder categories that we get in this framework. Okay, so now the next thing that I want to talk about is this thing called truncation. Okay, so if we start with a segment and we pick an element of our preorder, call it B, since omega is 0 and 1, in most cases it's going to be 1. Then I want to define this set, which means I'm going to truncate this segment by B. So what this means is I want to find all the nodes such that when I map them over via T and then I look at their image in the preorder, their image in the preorder, or in other words, their color is at least B. Okay? So if I take B to be 1, this is going to be picking out all the nodes that have color 1, or in other words, have color black. Okay. I said that. So here's an example. So if this is my segment, and I want to truncate this by 1, remember 1 means black, then that's going to pick out 2. Sorry, it's not going to pick out 1 or 2, it's going to pick out 3, not 4, not 5, not 6, 7, 8, 9, 10, skip 11, 12, and 13. So that's what truncating that segment is going to do. It's going to pick out those integers. And if we truncate by 0, we get everything. So I'm never going to truncate by 0 because it doesn't really do much. Okay, so what does truncation do to morphisms? So let's say we have a morphism given by a pair of maps, F1 and F0. Say this happens to be our F1 and F0. So I'm going to talk about an example before I say what happens in the general case. Okay, so we can truncate this guy and we can truncate this guy. So this is going to get 1, 2, 3, 6, 7, 8, 9, 10, 11, 12, 13, 14. So truncating the first guy, we pick up these numbers. And truncating the second guy, we're going to pick up 6, 7, 8, 9, 10, 11, 12, 13, 14, which if you'll notice, that's a subset of that one. Okay, and so the second guy is a subset of the first. So no, this doesn't have to happen in general. This is a very, very special case, but I'm just giving you an idea of what truncation is going to do to morphisms. To figure out what it does, okay, so before I talk about how to do it in general, so here's a proposition that says that if a particular guy gets truncated in the second segment, then it has to get truncated in the first segment. And this is just a reflection of the property that colors cannot increase. So in other words, you're not going to truncate something in the second segment, but not truncate it in the first segment, because that means that its color would have to have increased in order for that to happen. And that doesn't happen. Okay, so if we fix a positive integer n1, and we look at this sub-preorder category, then this map extends to a function. Okay, but we want to do a little bit better than this, because this is restricting ourselves to segments of this particular thing. We want to be able to truncate morphisms of segments in general that maybe necessarily don't have the same number of nodes. So how do we do that? So we need this injunction. We actually don't need the fact that this is an injunction, but it happens to be an injunction. So here set star is the category of pointed sets and morphisms of such, which means that they're functions that preserve the distinguished element. So what f does to a set is it just puts in a special guy called star. And given a function, then f of that function is just going to be this map that sends star to star, and then the rest is just f. Use the forgetful functor. So we have this adjunction. We're not going to use the fact that this is an adjunction in this, at least in this talk, but that's used at several points in the paper. Okay, so now I want to talk about how we can truncate morphisms in general. So to truncate morphisms in general, we need to talk about that. This is where we need the pointed sets. Okay, so the way that we do this is that if some I is truncated and it gets hit by, if some guy j gets truncated and he's hit by some I, then that's the I that we want to map j to. And if the j doesn't get hit, then he's going to go to this special element star. So in the previous example, it was conveniently cooked up so that everything getting truncated over here was actually hit by something. That doesn't have to happen. So here's an example. Here's an example of where that doesn't happen. Okay. So what's going on in this picture? Okay, so in this top row, what the subscripts denote are their place in the segment. Okay, so this is spot 1, spot 2, spot 3. In the second segment, what these subscripts denote are the image of the corresponding node in the first segment. Okay, so 1 means this guy's going here, this guy's going here, this guy's going here. This little star means nothing is hitting this guy. Okay, if nothing's hitting this guy, 4 is getting hit by 4, so on and so forth. Okay, this is not the fourth node in this segment, right? That would be this guy, I guess. Okay, so the subscripts denote where each one is going. Okay, so we can truncate both of these just as we did the previous ones. So if we truncate the first segment, we're going to pick up these numbers. And if we truncate the second segment, we're going to pick up these numbers. Okay, so notice 4 and 5 here. 4 and 5, these two guys that don't get hit by anything, we need to send these guys somewhere, and they're going to get sent to this special guy, star. So this is why we need to talk about pointed sets to truncate morphisms in general. And everything else that actually gets hit just goes back from where it came. Okay, so 1 goes back to 1, 2 goes back to 2, 3 goes back to 3.
So I can see this. So this truncation thing is going backwards, right? It's contrary. So if a guy has a pre-image, like, and it goes to its pre, why does it just have one pre-image?
Subjective.
You made it that way. Okay.
I didn't make it, or Rémy made it that way.
Okay. But we made it, okay. So everybody has at most one pre-image. So if it has one pre-image, it gets sent to its pre-image. If it doesn't, you send it over to star.
Exactly.
Okay, makes sense. That's a very general thing you could do. You could like So you're saying it's like a functor from, contravariant functor from like sets and injective maps to pointed sets where each guy gets into its pre-image or to star if it doesn't have it. Yeah, that's something I never heard of. That's really cool. Yeah.
Right. So now I want to let E and epsilon be a pointed set. So the epsilon is going to be the special distinguished element of E. And I want to consider this composition of functors. And I want to denote this composition as E sub B epsilon. Maybe I'll just call this E sub B, I guess. And I want to look at what this composition does to segments over omega and morphisms of segments. Okay, so first let's talk about what it does to segments. In other words, objects, okay? So omega, still a Boolean preorder. And I want to take my set E to be AC, GT, and this little epsilon. Okay, so for AC, G, and T, you should be thinking about, of course, the nucleobases from the very start. Okay, so if we take a segment, for example, this guy, and we apply this composition of functors to it. So first, we're going to truncate it, which is going to pick out the spots where all the black nodes appear. And then we're going to map those into this set, this pointed set. And so we're going to get sequences that look like this. So, I mean, the notation is very visually suggestive. So we got 3 guys here, so we can take 3 guys from here, 4 guys here, 4 guys here, 5 guys there, 5 guys there. And then the white white patches, they get thrown out. Nothing happens there. So we basically get tuples that look like this.
So except for those epsilons, these are like ways that you could have. I don't know what's going on. I'm going to think they call these black guys the ones that are expressed.
Yes, exactly.
So they're like ones that you're actually going to do something with. The white guys, you like, are the guys that you're going to ignore. So the ones that are expressed, they could be A, T, C's, or G's. Why do you want to let them be epsilons too?
Well, I don't necessarily want them to be epsilon.
Well, why do they get?
They can be epsilon.
Stop excusing yourself. I'm just using you in the general sense.
So in an example in a second, there's going to be a particular case where some guys have to be epsilon.
Okay.
That's where.
Okay, I'm just curious about this.
Yeah. So what does this composition do to morphisms? So I think this is a different example from earlier, but in any case, so here we got some guys that are not being hit by anything. Okay, so this guy is not hit by anything, but he's white, so we don't care about him. These 2 guys were truncated, so when we truncate by 1, these guys get picked up, but they're not hit by anything. This guy is also not hit by anything, so these are going to be the guys that need to be expressed as epsilon. So this is going to give us a set of maps that look like this. Okay, so again, I mean, notice we got 3 here, 4 here, 2 here, 3, 4, 2, 3, 4, 2, 3, 4, 2. But over here, okay, we got 5, 4, and 1, 5, 4, 1, 5, 4, 1, 5, 4, 1. But notice for these 2 guys, these little stars that don't get hit, these are forced to be epsilon. Okay? This last guy is also not hit. He has to be epsilon. The rest of them can be whatever, but we need epsilons there, and there. Was that?
Sort of, yeah.
Okay. So that's basically what the epsilon is for. Is when we have maps like this, where not all the points.
Epsilon means like leave that spot blank.
Yeah, sort of, pretty much. It's sort of like a, I don't know if I want to say placeholder, but
So leave it blank, or you could put in anything you want.
I don't think we can put it in. I mean, not for this particular map. Like, if this was, like, I can't make this be like A.
So why are these identities other than adding on the epsilons?
Why are these?
Or what identities?
These maps, they are the identities except for adding epsilons.
Oh, yeah. So like, oh yeah, no, I guess they don't have to be, but they have in these examples, I guess they are. I don't know if that has to be true in general though. But yeah, I see what you're saying. Like, this is staying the same as this. The only thing that's actually changing is you're putting in two epsilons here and then tacking on the epsilon here.
Is the functor gives a set of functions in E, and the functor is just composition with the map of segments.
Can you pop back to the definition of big E on morphisms? So,
The definition of big E on morphisms?
oh, you didn't define it. You're just
Yeah,
giving us
just doing
examples.
this.
Okay. So anyway, okay. Well, when we see the definition of, or if we ever see the definition of E on morphisms.
Probably not.
Okay. If someone saw the definitions on morphisms, they would probably see that was required. the feature Christian said. I think that's what you were saying.
Or yeah, I think it's composition with the inclusion of the truncations.
To the extent that I understand you, yeah, I agree.
Yeah, could you go
It's
back
like a
real quick
composition thing. But
to the composition of the functors?
Oh, yeah. It's
We actually did, that's the definition. Okay, so you did define it, but it's rather abstract. But that does it. So what Jade was saying was talking about this second part. That a map of sets, pointed sets, induces a map between their sets of functions to E, like pullback pre-composition. So that's making that thing happen. You did show us the definition.
Well, I didn't put it in the definition.
Well, there it is.
So I
It's
think
really tiny.
It's full of symbols, and
unofficial.
it's very small, but it's all
definition.
there, yeah.
Okay, so I think the next thing I talk about are codes. After this slide, yes. Okay. So yeah, so a cone roughly looks like this. Most of the cones that I'm going to consider are going to be over discrete diagrams, which means that there's no morphisms between the objects on the base. But you could have stuff going on down here, which would translate to stuff going on over here. Yeah, so I think most people have seen. I mean, if I'm not going to define an injunction, but then to define it, I don't know why I even have this slide in here. Okay, so the reason I want to talk about cones is because there are these things called exactly distributive cones, which are important for what I want to do next. Okay, so omega is a preorder, B is some element of that preorder, A is going to be a small category, T is going to be some segment over omega of a length n. And I want to consider a cone whose apex is tau inside this preorder category. So something like this. So this cone again happens to look like it's discrete since I got nothing going on down here. So something like that. So I can truncate this cone, and that's going to reverse all of the arrows. So that's why I'm working this particular preordered category. Because if I do it for arbitrary segments, then we have to do the stuff that I just talked about. But if they all have the same length, then I can truncate all of them, and the arrows just flip around. So this was like the first example where truncating the second guy just conveniently happened to be a subset of the first guy. Okay, so we get this co-cone in set. And then we can look at this map, okay? So here we're utilizing the epi monofactorization in set from this guy to this guy. Okay, so all the guys in A, in the diagram A from the previous slide, they all have maps going into tau. So the co-limit of A is going to have a map going into tau. We can truncate that guy. And then we're going to map this guy through the union of all the guys in A and truncate all of them. So M is monic and E is epic, and this is using the epi monofactorization set. Okay, so the particular cones that we care about. So a cone is said to be B-distributive if this monic guy is also epic, and then it's going to be exactly B-distributive if this M guy, which is monic, is epic, and this epic guy is also monic. So in other words, this is an isomorphism. So if this happens to be an isomorphism, then we say that the cone is exactly B-distributive. And these cones are nice because they're going to let us do some stuff in a little bit. So this is probably one of the most sticky things to understand in this whole talk. So let me give you some examples of exactly, or let me give you some examples of these cones. So okay, in case you forgot, omega is still the Boolean preorder. So I want to take a cone over the discrete diagram with just 3 objects, and these are going to be segments of length 18. So here's an example of a cone. So this is a sloppy way of drawing it, but so these guys, these are all the same. This is our apex, and this is our diagram A. It's these. This is a guy. This is a guy. This is a guy. These black nodes are not the same as these black nodes. It's just an unfortunate abuse of notation. So okay, so we can start truncating stuff. So this is our tau, this is our apex. If we truncate this guy, we're going to pick up these numbers, which you can check if you really want. And then if we truncate the co-limit of A, well, A is just this discrete diagram. We're going to pick up these guys, where I'm putting the primes to denote we're picking up duplicates, and that's easy to. So like here we're picking up, let's see, 1, 2, 3, 4, 5. So we're going to get a 6, a 7, 8, and a 9, and we're also going to get another 6, 7, 8, and 9 from this guy. So that's why I've got a 6, 7, 8, 9 here, and then I got 6, 7, 8, 9 primes, and likewise, this guy and this guy, these are duplicates, so that's going to give us this 10, 11, 12, 13, 14 with primes. Okay, so this map, in this particular case, it's only epic, but it's not monic, yeah?
So the underlying orders in objects don't need to be total orders?
The omega, is that what you're talking about?
So no. In the an object is it T0 and T1?
An object, it's
And 0 and 1?
Yeah, and yeah.
so an object is like one of these guys up here. So it's a T and a C.
So then, is this the object, this co-limit?
No, this is just a set. This guy.
It's a set.
This is
Oh,
just a
okay.
set.
It's of the truncation.
Okay.
The map is the map induced by the universal property of the co-limit that we're talking about.
Yeah, exactly. Okay. So because this is epic and not monic. This is not an isomorphism. This is not an example of an exactly distributive cone. So there's a couple ways that we can fix that. So one thing we can do is we can just not have this overlap here and this overlap here. So in other words, if I make these guys white or these guys white, as well as making either these guys white or these guys white, that would fix the problem, because then we wouldn't be getting these primes here. Or another thing we can do is we can identify this patch with this patch, this patch with this patch in the diagram A by adding some more stuff. Okay, so this is This is only distributive. This is not exactly distributive. So we're not gonna care about cones like this. This is just an example. So a better example would be something like this. Okay, so now there's no overlap between these guys, which you can see. So that map is going to be an isomorphism. Here's the way I should really be drawing it. This looks more like a cone. So the point here is that if I have a cone over a discrete diagram, such as this, then I don't want to have any overlap between the guys in the base diagram, between the black pieces. And every black patch up here needs to appear exactly once somewhere down here in the diagram A. Okay, if the diagram A is to be discrete. It doesn't have to be.
So this is like splitting up work?
Yeah, it's sort of like it's like isolating each one of these patches. Another example. So this is what I just said verbally. Here's another. So this guy is also exactly distributive. Okay, so if you look at this guy, and this guy, notice they all have this little piece, this black piece in common. Okay, so normally that would cause a problem, but if we identify all of them down here, so this is the diagram A, this is the apex, then this cone is also going to be exactly distributive. So yeah. Okay, so what can we do with these cones in this framework? So some things that we can do are duplication. We can do this thing called Crispr, which sounds like a brand of easy bake oven. We can talk about transcription and translation, mutations, and inversions. So these are the things, these are going to be my examples. Okay, so let's start off with duplication. Okay, omega is still the Boolean preorder, and E epsilon is going to be this set ACGT epsilon. Okay, so we got these two morphisms that live in segments over omega, and the subscripts here are denoting the images of the little black things. Okay, so we got these two morphisms, and if we apply this functor E to either one of these morphisms, it's going to be an identity that sends any word of length 3 to itself. Okay, so for example, ATG goes to ATG. Yeah.
So the truncation throws out the white guys
Yes.
and keeps the black guys, and this is sort of how it colors. So it's sort of like you have like 3 letters that you're wanting to express or something like that. And you have like 6 places you can put them. And you have to pick which. Well, F1 says we're going to pick the first 3 to express our A, T, and G. And throw out the last 3. He says we'll throw out the first 3 and keep the last 3, but then they have the same effect.
All right. So now we have this cone, which is exactly one distributive. It's over a discrete diagram. There's no overlap, and every black patch up there appears somewhere down here. So because this cone is exactly one distributive, this is, at least in this talk, this is the example why we care about exactly distributive cones. This particular map from the apex to these 2 guys down here is going to be invertible. And I guess that's pretty easy to believe, because here it's like we got a 6-tuple of guys from our set. And yeah, of course that's going to be in bijection with a 3-tuple here times another 3-tuple there. So the inverse, just flip the arrow around. That's the inverse. So if I take this guy and I pre-compose him with the previous guy. Okay, so what this map is doing is it's taking a word of length 3 and another word of length 3. It's just going to stick them together. Exactly what it looks like. And here, I guess I'm going to start ignoring ignoring the parentheses. You could put parentheses here and parentheses here if you want, but also remember we can merge patches, so it's fine to write it like that. So this just takes 2 words of length 3, puts them together, gives you a word of length 6. Okay, so that map, we apply this functor E1 to it, that's going to give us this map. So this is where the notation should look visually suggestive. We got this guy going to here, and this guy matches up with this guy, okay, except for the subscripts, but that's fine. So if we compose these guys, we're going to get a map going from here to there. That takes...
Buttons use colimits. Why are there Cartesian products showing up?
What's that?
So I thought earlier in the definition of these maps you use colimits or colimits. So why is there a Cartesian product here?
So the only place I used, you mean when I was taking the colimit of the diagram A? Is that right?
For the definition of distributed.
No, I'm not sure what you're asking.
I guess I don't understand what the map view is.
What the map mu is? Oh, I'm not telling you what the map mu is. I'm saying that because the cone is exactly distributive, the map mu is going to be invertible.
Why is that? Well,
I'm taking details and sweeping them under the rug. Yeah, so this, in general, this map mu is not going to be invertible. It's because the cone is exactly distributed that this map mu is invertible.
and there's probably some map mu that I could figure out if I looked at one and figure out what mu is.
Yeah, I mean, an exactly distributed cone corresponds to a way of like splitting up the black patches into these separate ones, and then you could just, if you know that that bijection exists, then you can construct this map from the product of those splits back to concatenate.
Yeah, I'm not telling you where the map mu comes from. I'm
Okay.
completely bypassing that. I'm just saying this map mu exists, and it's going to be invertible in the case the cone is exactly distributed. That's all that I'm saying. Okay, so if we compose these guys, we get a map that looks like this, which does exactly what it looks like it's going to do. It's going to take a word of length 3, and it's going to concatenate it with itself. So something like this.
So what is the e's?
Those are the set of functions from the truncation of your segment into the point itself.
Yes, this is a composition of functors. You truncate it by B, and then you map into E, roughly speaking.
And E is the set of A, T, D.
Yes, E is the pointed set, A, C, T, G, epsilon. Okay, so that's duplication. So another thing I want to talk about is Crispr. So Crispr stands for clustered, regularly interspaced, short, palindromic repeats. So what Crispr is used for is there's another enzyme by the name of Cas9, and these two guys work together in order to locate and to cut specific strands of DNA. So this picture is really, it's honestly not meant to be funny. So when I was making these slides, I was like, okay, I want a picture for Cas9 and Crispr, and I typed that into Google Images. And you get pictures with all sorts of stuff going on, with millions of other little things. And I didn't want to put those in because I figured that would just confuse everyone, myself included, even more.
Where's this?
Whereas this, yes, this is a very simple analogy where Cas9 says, I want to find Crispr, and I said, okay, I get the point that this is trying to make. So, yeah, so this is basically what I just said. So these two guys work together, and we can use these guys to, or I should say people supposedly, use these guys to edit genes. Here's a very overly simplistic picture of that, cutting some DNA with a pair of scissors. And according to the great and all-knowing Wikipedia, this is used by biotech companies, and it's a research tool. And mean, you can do stuff like maybe treat diseases and all sorts of other things. So on Tuesdays, I have a 6B, Math 6B in the Life Sciences building. And a couple weeks ago, I was walking there, and I happened to just be going through the hallway, and I looked on the wall, and I saw this picture. So I don't really know what's going on here, or what this mouse is doing, but I saw the word, and I was like, oh, it's the thing from the paper. So yeah, this is a real thing, I guess. So okay. So what does it actually look like in this framework? Okay, so suppose we have a segment given by this. ATC, GTC. Well, I shouldn't say this is a segment. It's a segment of DNA, but it's not within the context of the framework yet necessarily. Okay, so suppose we have something like this, and I want to rewrite the CGT in here, okay, as TTC. So in order to do that, first, so yeah, that's what that would look like. I want to somehow go from that to that. So first I need to select the CGT inside this word of length 6, and then I want to somehow replace it with a TTC. So how are we going to do that? Okay. So the initial segment ATC, GTC, that's going to live in here, okay? If we ignore the brackets, which we can. Okay, so in other words, AT is going to be here, CGT is going to be here, C is going to be there. And you can sort of guess what's going to happen. We're going to throw out the CGT, get our hands on a TTC, and then we're going to put those guys together. Okay, so TTC lives in here, okay, because we don't care about these white guys, okay? We only care about the black patch, and TTC is going to be in there. All right, and I want to either cleverly or not so cleverly pick this to be my map F. Okay, so this is going to take a word of length 6, and it's going to throw out 3, 4, and 5 after I hit this guy with my E functor. And the other guy is going to be an identity. So this is the map I'm talking about right here. Okay, so this is where my initial segment ATC, GTC is going to live. And I'm going to have my TTC in here. I'm going to take my initial segment, I'm going to throw out the CGT, and the TTC is just going to come along for the ride for now. And then another exactly distributive cone is going to come in and save the day. So this is the exactly distributive cone. No overlap, and every black patch appears exactly once in the base diagram. So this map mu, which I'm not telling you where it comes from, but it's going to be invertible. So this is the map mu. Goes from here to here, so then the inverse just... So the reason why I'm sweeping these details under the rug, I thought it would be, I don't know, very believable, I don't know if I should say that tuples like this are going to be in bijections with tuples here and tuples there. Maybe in hindsight, it wasn't good to sweep those details under the rug, but
Yeah, it's very believable that there is that. I guess part of what Jade was wondering about is you were talking about. Let's see, you were talking about colimits for a while,
Mhm.
and then now this product is snuck in. So
Yeah.
it just maybe there's. Is that just because there's a contravariant functor
Maybe.
running around? Maybe it's just
Because
you're taking a co-limit of a something. Whatever, co-cone?
yeah, the distributivity you expressed in terms of co-limits. You said this is going to be exactly be distributed if
that composition of maps is an isomorphism.
yes, and that composition had a co-limit.
Uh-huh.
And then what Jade is probably asking is whether this product on the right hand side came from, where did it come from? Did you apply a contravariant functor and it turned co-limits to limits or something? Maybe this epsilon?
Maybe just, I mean, that E function
Yeah,
is
that E.
contravariant, so
Mhm.
maybe that.
You said it's not
Turn.
obvious that
Maybe.
map exists. I just don't see why it's obvious, or I just like the intuition behind what that matters. It's like it's sort of on this.
The intuition is that if you have like 6 little guys, black guys there, each one of them is going to get labeled with, that E means the set of all ways of labeling those 6 guys with letters. So if I have any way of labeling those 6 guys with letters, I can turn it into a way of labeling these 3 guys with letters and those 3 guys with letters. I just use the same letters. Maybe you're going to do an example.
Okay.
Yeah,
Yeah.
well, sort of. Yeah, so the inverse, you just turn the arrow around and you get that. So we got this map together with the map from the previous slide. I put them both up there. These are the 2 maps. So again, yeah, so if you notice, this is exactly that. So then if we combine these, we can go from here to here, and then that map will do exactly what we want it to do. So, so this map, it's going to go from here to here. So we're going to take this guy, we're going to throw him out, and we're going to replace it with this guy, which is exactly what we wanted to do for those sequences of letters. So if we plug in this pair, out comes what we want. So we're replacing this guy with this guy inside of there. Okay, so next, transcription. So if we start out with a map of pointed sets, then we're going to get, I should say this map F is going to induce this natural transformation. So we can truncate and then map into A, and then we can apply this map F, and then that's going to be the same as truncating and then mapping into B if we compose via F. So this map F gives us this natural transformation. So I want to define a bijection of pointed sets. So I'm taking, so now I'm going to call this set A, I guess. It's the same set E as before. But now I want my set. So now the set B is going to be ACGEU. Okay, so remember in RNA transcription we had U's flying around instead of T's. So that's going to be my set B. And I want to define my map F by how RNA transcription works, which is like this. So every time we see an A, it's going to give us a U, so on and so forth. So the previous natural transformation, that's going to give us this map, which is going to send words that live in A to words that live in B. So stuff like this, okay? So this word's going to get mapped to that word based on how the map F is defined. So A goes to U, G goes to C, so on and so forth. And this should be the picture from earlier. And I tried to pick the letters that sort of match the picture. So here you can see we have AAG, and then in the transcription, that's going to UUC. So AAG, UUC, so on and so forth. So TGC goes to ACG, GTG goes to this AC. So here's how we can model transcription. And then next, we got mutations. So the way that we can talk about mutations are going to be, so E is going to be the same set as before, and we can take the product of this set with itself to get another pointed set, where the special guy is the pair epsilon, epsilon. Okay, and then this product comes with projections to each component, P and Q, and then just like in the previous example, these are going to induce natural transformations. So in other words, we can truncate into here and then map into there via this map, and likewise for Q. So what this is going to do is it's going to give us a span, and this span is going to represent all the different ways that one strand of DNA can be related to another. So, for example, so this is just these are just a couple of maps, and this is what they would do. Okay, so these guys look like this because these are now elements of E times E. So what this is doing is this is taking all the guys, I believe, in this, looks like the second row, and this guy is taking all the guys in the first row. So in other words, the first component, these are really pairs, okay? And the second component gets projected to there. So it's just a relation between the guy on the right and the guy on the left. Okay, and the last thing I want to talk about are inversions. So if we start with a positive integer n, then we consider this map. So before you start thinking, what is this map really doing? I'll give you an example. It's just going to reverse the ordering. So yeah, that's what it does.
Notice whenever you make a bunch of displayed equations right on top of each other in LaTeX, it makes a bigger space between the first two.
Oh, wow.
Can't stand it.
Why?
I don't know, it always does that. Some bug in LaTeX. I see it there.
You gotta use a line.
Or gather, yes. A line of
Yeah,
gather
but-
is going to
Anyway, never mind.
cut it off.
You should have solved that problem.
I know.
Clearly.
You obviously
Okay.
didn't put enough work into it.
So if we start with the segment, then we can pre-compose and post-compose with particular maps, RVN0, RVN1. So all this is going to do is it's going to take a segment and it's going to turn it around. So for example, if we have a segment with 9 nodes and 6 patches given by this, and we pre-compose with this RV9 and then post-compose with an RV6, it's just going to take this guy and it's going to flip him around. So we're going to get this. And I want to denote the inversion. So yeah, we're going to call this guy the inversion of that guy. We're going to denote that with a little dagger. Okay, so this induces an endofunctor from the category of segments over omega to itself. Okay, so this is our functor E from before, and the map from the previous slide is going to induce this natural transformation, which is just going to take a segment and it's going to invert it. So what this is going to look like, if, for example, we take this to be our map, and we take this to be our sequence of letters, A, G, T, A, G, C, it's just going to take it and it's going to flip it around. So C, G, A, T, J. It's the same thing, just in reverse. So one application of this, if you remember from my spiel about how DNA is duplicated, another example, okay. So this guy at the bottom, this DNA polymerase that works on the lagging strand, he needs to read the segment of DNA backwards. So inversion can be useful for taking that segment and flipping it around, depending on what you're trying to do with this stuff. Lagging strand, it should be strand, not strand. Okay, so I think this was like the end of the third section in this paper, and the next section started about something completely different. So it's very anticlimactic. This is the end. This is where I decided to stop.
Yeah, I like that a lot because I looked at this paper and I just didn't get it. There's a lot of constructions and I just didn't put enough energy into it so I could understand what they were all good for. Now I feel happier about this paper that he's like using math to more or less capture what's really going on. That's what the point is. Any other questions, comments, complaints?
So it seems like this is all setting up like a category theoretic language for talking about these processes. And my question is, does the author then like use that language to deduce something interesting about the processes that's like?
Well, I guess that depends on what you mean by interesting.
That's a good answer.
So yeah, he does. I mean, he does a. So this is like the first. This is the first half of one of 3 papers. So he does a lot of stuff with this. I can't even really begin to give you a good answer as to what the rest of that stuff is, and much less as to whether or not if it's interesting. So I guess one thing that you would want to try to do with this framework is to actually try to answer some questions in biology that are actually difficult. I don't know what those questions are, much less whether or not they can be, if anything can be said about them within this framework. So, I mean, that's a very vague and hand-wavy answer to your question, but so like some of the things that I mentioned on like the first couple of slides, like he talks about like genetic linkage and sequence alignments and haplotypes and genotypes and a bunch of other stuff. Like I don't know what that stuff is, to be perfectly honest with you. So that stuff might be interesting, maybe.
Is this all his thing?
Yeah, this is all his thing. I did none of this. Nope. Yeah, this is all his.
I mean, like,
So I mean, you can imagine that with this stuff, you could like come up with a good computer programming language for searching through possibilities in genetics. You know, what are all the ways that you can, so with these certain rules, you can generate some string of nucleobases, and then like that might be something that you were interested in biologically. This could be like the general sort of programming language for answering genetics questions. But I sort of don't think Tuyéras has done that yet. I think I would have heard about it.
It's cool how easy it was to represent those fundamental processes, mutation and inversion and replacement and that stuff. And now I'm wondering if he goes in to explore, biologically I'm sure there's some deep interactions between those processes and how they're linked together, and then I'm wondering if they're going to end up corresponding to the categorical intertwining, because they were pretty easy to express in his framework, but then they were each kind of like, I'm going to define this function that does the inversion or whatever, you know, just a function out of nowhere. So I'm wondering if there's some nice, translating the nice biological relationships into the method.
Yeah, he later, so I don't even know if this is going to make any sense, but he later goes on to show that certain constructions and whatever, but I don't need it. Certain constructions involving what he calls pedigrads, which are they're basically functors from categories like seg omega into another category, call it C, that in a very nice way maps cones from seg omega into cones of C, and he shows that they satisfy a universal property of some sort. I don't remember what it was, and I couldn't even begin to tell you, but yeah.
One comment is that segments of omega is almost a common category. So that's kind of interesting.
Yeah, it's probably one of those shaped common categories that we
Who's doing it?
for Petrinets, it was like a common category.
Oh,
Yeah.
Yeah, sort of like. But, yeah, anyway, I think it's really an open question yet how useful all this stuff is. And I haven't heard like lots of people say this stuff is like solving all sorts of wonderful problems. It's an initiative to try to use category theory to figure out the math underlying what DNA does in cells. So category theorists can easily like that kind of thing. Because there's all these things that DNA does, and it would be very, if you're a category theorist, you just want some crisp mathematical framework that says what all those things are. Everyone who's not a category theorist will say like, okay, great, but now what can you do with it? You know, are you gonna like do something that will wow molecular biologists? And as often with these initiatives, it takes a lot longer to get to the second part than the first part.
I think part of it is that I don't really know what any of the deeper constructions are in category theory. So to me, it just looks like a language that's, you know, similar to permutations or arrays or something like that, that doesn't appear to have any additional structure. But I mean, I figure that once you pull it into the category theory world, that there's a bunch of like theorems and results in category theory that, you know, then can be interpreted to apply to that. And I guess that's kind of what I'm wondering, like, you know, once you have it in this language, like what does category theory say about it?
I'll have to go read the rest of Tuyéras's papers to figure out the answer to that. Yeah, so in good cases, and I try to write papers that are good cases, you take some problem, you formalize it in category theory, and then like use always a mixture of category theory and the specialized math or whatever problem you're studying to prove stuff. But it's never just purely category theory, but as far as I know, it solves all your problems. So like I do like a mixture of differential equation theory and category theory, like Kenny and I in the paper. It's right next to this
Yep.
on the journal, Open Markov Processes.
Well, not this one in particular, but the one of the other ones is, yeah.
Yeah. Anyway, so
And then-
sometimes, so in other words, sometimes it really does what you're hoping for does really happen, but I just don't know about it for this specific
Yeah, I mean,
initiative here.
that's
And
not to
sometimes
say.
it doesn't happen. So right, so there are lots of people who try to apply category theory and it's sort of, you know, like, you don't know really what happens to it. Yeah, it's just like you set it up and then they like say, I'll go do something else. So, because it's very hard to carry through.
Yeah, and it's not to say that he doesn't do something really cool with all this stuff, because again, like I stopped like in the middle of the paper.
Yeah,
Just
well,
with
since some
these
of us sort of
examples.
know David Spivak, we like email Rémy Tuyéras and just ask him, so what became of all of this? Or do you have some big plans for what to do with this next? That's the quickest way to find out, I guess. Okay, great.